what we're going to do now is do two things. One is I am going to introduce your research research paper project, and then we are going to review for the lab practical. So if you go down here to remote week 9, which is where we're at right now, you will see like, oh my God, there it is. Introduction to research paper and presentation, choosing a topic. So the first part of this is just figuring out your topic. I'm not asking you to do, so over the next few weeks, what you're going to want to do is think about what do you want to do this project on. So let me talk a bit what the project is so you have a better sense of what you want to do. Okay, so this will be similar to the literature search homework, but done at a more rigorous level. You will write a research paper on a current topic involving human physiology using the scientific literature to support your discussion and summary of our current scientific understanding of your topic. Presentation and paper should be on a topic that interests you and can be written to inform us about the latest application of a topic. So I mean, one of the things that was really cool last semester was people wrote about things they really cared about. Some people wrote about kind of medical conditions that they or someone they knew were like dealing with, so they could understand it more themselves and share to other people so other people would understand what's going on. Some people wrote about more social issues around different kinds of addiction or things like that. Some people wrote about all sorts of stuff. I would say this is going to be way more interesting to do for you and for everybody else if you write about something you're genuinely interested in, because then it's actually sometimes, I know for me even sometimes it's kind of nice to finally have an excuse. I wanted to look at this stuff, but I'm just so busy in my life. It's just in triage of trying to just stay afloat. I don't get to actually look at the stuff I actually genuinely am interested in. So this kind of gives you, an excuse. You've got a motivation like, okay, I'm really wanted to know more about this. So, and I have to do something to get my grade in the class. So let's study this. So this can be a really kind of exciting thing to do. So, it can be exploring if popular beliefs are supported by scientific research. There's a lot of kind of word of mouth stuff about health or this or that, but then you're wondering, like, is that real or not? This is a time to go deeper and see, kind of like we did with the literature search homework. Like, do oats really seem to help? It's like reality, it looks like they do, although it's a little not quite, you need a lot of them, and it's just one of many different things, but it's nice to go and see what is real, what's not real. So the initial, and this is the other thing I put here. So two things. One, it's going to be worth 75 points for the final lab grade, which means it's as much as like this exam you're taking on Thursday. It's worth a lot of points. And it can be done individually or in pairs. If you do it in pairs, you are going to have to like do an accounting of like, who found what resources, who was responsible for what parts of the write-up in general. I want to have some sense that you both contributed, that it wasn't just one person that just wrote on someone else's coattails. If you do it in a pair, pick somebody you work well with. It can be really fun. I'm sure you've had this experience, and I can say just in this class, watching people work in groups, like sometimes it's like the best time ever. Kind of people really dig working with each other and really inspire each other. But sometimes it becomes a nightmare, and it's So you have a choice. If you want to just do it on your own, do it on your own. If you want to do it with somebody, that's great. Um, I would just say make sure you pick somebody that has a similar kind of work ethic to you, who's going to like take it as seriously as you, and is going to like feel like, oh, this is kind of fun and we're synergizing, versus like, why did I get into this? So just kind of putting that out there, because yeah, I mean, over the years I have seen like people just again have the most fun ever on joint projects, and then I've also seen people just come just being miserable. So da, da, da. So initial instructions, this first part, this is, well, all you need to do now. There's no grade to this. This is coming up with an idea, starting to brainstorm. And then once you brainstorm, though, you need to make sure there actually is stuff out there that is going to give you enough resources to write a good paper. You might have an interesting question, but then you find nobody's really done research on it. There's nothing really. I want to write this paper. And then when I try to go into PubMed, it's like, oh my God, there's nothing. So you need to make sure you pick something where you're not setting yourself up to fail because there's no research on it. So this first part here is think about topics, go into PubMed and see if there is actually enough research out there that you could actually write a reasonable paper about it, supported by scientific literature. And then once you settle in on it, make sure it's appropriate to our class. This is a physiology class, so it should be relevant to human physiology. And then once you have something interesting to you, appropriate to our class, and has adequate relevant research so you can actually support your paper with scientific journal articles, write a brief 1 or 2 sentence proposal. So this is, don't write up the paper, because I might say, no, that's not good. Or I might give you like, how about if we shift it a little? But this should be, most of the time I say that looks good, go for it. But you do, this first part is just sometime before, in the next week and a half, what I would like you to do is figure out what are in, it could be 1 or 2. If you have a couple, you could write a couple, but give me your proposal for the thing that you want your paper to be about. And again, don't start it. We're not starting. I'll give you way more information about the details about how to write it and what the expectations are. This is more just starting to think about what you want to do. You are going to also do a presentation. You're going to make a PowerPoint presentation, and we're all going to watch each other's PowerPoint presentations in the last week of class. So that is actually really nice. People learn a lot from each other here. And people usually do some really nice presentations. Here's things people have done, physiological effects of social isolation. That was, I think, something that was really interesting. Somebody came up with as we started doing the shelter in place, health benefits of drinking red wine, oat bran and cardiovascular health. You've seen that one. Exercise and hypertension, creatine phosphate and exercise, benefits of energy drinks, risk of steroids, drinking during pregnancy, heart disease and exercise, psychedelics and mental health, exercise in the immune system, alcohol in the brain. It can be anything as long as it's tied into physiology. So again, I encourage you to pick something that you like. There are many beliefs and misconceptions about physiology and health. You can go explore scientific evidence to either support or not. Again, like oat bran and cholesterol. Here I said, things that will make you lose points on a project. not using scientific journal articles, using unreliable websites such as Wikipedia and stuff. So this, when you're trying to figure out, is this a good topic? Make sure that there is actual scientific research. If you just find stuff on, bloodtypes.com and like why eating this food is good for you because of your blood type or whatever, and it's like, but is it real? Like anybody can make a website that says anything. Wikipedia, we could, in fact, the more you know about the world, the more you realize Wikipedia is useful for a very first approximation and then goes downhill really quick. You really need to make sure your paper is going to be grounded in kind of primary research. So make sure as you're kind of looking at the literature that primary research exists about the topic that you want to write about. So sometime in the next week and a half, you just submit one. If you're working in pairs, you only need it, just one person in the pair can submit it and that'll be fine. Just make sure you have both your names on it so I know that it's two people involved. Are there any questions about what the expectations are for the big project in general, which is basically oat bran homework on steroids? And then the PowerPoint presentation is like a 10-minute presentation of your findings that we will then share with the class.
I have a question just to clarify too, because the examples that you gave, at least listed there, are a lot of like independent variable, dependent variable, something upon something else and investigating that, but you could also do it on like multiple sclerosis or something like that, like a disorder or a disease.
Yeah, although it would be, as you're doing that, it would be about like, what are the current thinkings about what is going on and how to treat it and stuff.
Yeah,
right?
okay.
It still has to be connected to research, right? And research usually is about a, relationship between independent and dependent variables. So I don't want something that is, just you go into a book and read about something and do a book report. It has to tie into the research literature.
Got it. Thank you.
Professor, one question, me. Can we use peer-reviewed articles from the university, or has to be just PubMed?
What? No.
From the library, from the COM library.
Right. So, peer-reviewed, yeah, COM library has, through some of its online sources, has access to a lot of peer-reviewed journals and stuff. Yeah, so you can get into, PubMed is kind of one of the main search engines, but you can use, I forget, in Google Scholar, you can use, I forget the one that our library has one, what is it called?
Called EBSCO?
Yeah, something like that. So, and again, if you are trying to, for this first part particularly, all you're going to need are the abstracts to get a sense of what the research says. But as you get deeper into your paper, if you want full texts of things, just ask and I will make sure you can get a full text of anything without paying. Some of these things ask you to pay for things. So never ever pay for a PDF because we can get it for you for free. So any comments, questions about that? And it'll just be going kind of in the background. So you've got like the next 2 months ultimately to get this thing done. So, all right, so now we are moving on to the lab exam. So next class, when we get to lab, there will be a lab exam and it will also be a proctorio exam. It will be different from the first exam in that there'll be more, pictures and stuff rather than just all kind of questions. They're more likely to see graphs you have to analyze or pictures of test tubes you have to interpret what the heck happened in there or something like that. So again, the lab practical, it's, if we were in, in person, there would be actual stuff you could poke at. But unfortunately, we'll have to do our best with online. The lab exam will be on everything from the very beginning, including like what is an independent and dependent variable, which some people are still having some issues with, although a lot of people are getting more and more solid with. And then it's going to just go through every lab that we've done, and it will be looking at the main concepts as well as the techniques. You should know, like, if you see a test tube that had been filled with some blood cells and some solution and spun down, and you look at it, you should be able to, like, what happened in there? Just, you should remember, what does it mean to have a red supernatant or a pellet or this or that? So it's not just going to be kind of ideas. It's also going to be the practical stuff that we've been doing.
So, one question. So is the exam open lab notebook? Like,
So it is open. So your lab notebook, yeah. Your lab notebook is your part of the reason you have been hopefully doing a good lab notebook is it is gonna make this exam way easier, because it is okay. It is not okay to put crib notes in your lab notebook. The lab notebook should just be a record of what you did, what you found. Your discussion can talk about concepts and stuff, obviously. It should. But you shouldn't just like have like, basically like notes. It shouldn't just be notes in there. It should be a record of your lab work. So putting that out there, but if you've done your lab write-ups well, it's going to have everything you need, because your discussion is going to bring in all of the relevant concepts that are important to interpret what happened. So you are not allowed to have, well, that's wild. That's trippy. It's like there's a hole in my notebook. Anyway, the lab notebook is okay. Other notebooks are not okay. So it's just your lab notebook. That being said, though, your lab notebook, and I added a little thing before Thursday morning. I would like you to scan your table of contents and send that into me as well. So if you look at the, in fact, I can show it to you. Where is it? Here it is. So here, where it says lab exam, which is going to be October 22nd, lab exam 1 does not exist yet, so it's not published. But this it should be visible to you right now. Submit PDF of lab book table of contents. So I just want to, make sure you've done that and get a sense of how your thing is put together. And again, that will also help you if you have not been good at it, to make sure you have this so you can like find things in your lab notebook. So in terms of the actual exam, I brought up the PDF here. Yep. In fact, I should start with let me I still dig that. All right. Objective and independent and dependent variable. I would say for each of the experiments where it's relevant, you really want to make sure you know what was the question we were asking. Most of you have got that at this point. There's still a few people where, as I finally did finish grading everything, at least the ones that have been turned in, and there are still some people who are still kind of lost on what were we doing here? So you really want to make sure you're solid on what was the independent and dependent variable for each of the experiments we did. And then in more theoretically, what is an independent and dependent variable? Like if I was trying to figure out like which cat food is going to give my cat it's the most luxurious fur, and I feed him like this kibble and that kibble and give him some this Weruva wet food pouches, and I give him all these different kinds of foods, and and then I for different periods of time, and I like kind of assess like how silky is this fur. I'm going to rate the fur silkiness from 1 to 10. And I figure out which food seems to result in the silkiest fur on my kitty cats. What would be the independent and dependent variables there?
Independent would be the food, and then the smoothness of the fur of your cat would be the dependent variable.
Yeah, so the silkiness of the fur would be like dependent. I'm seeing how that changes in response to as I manipulate my independent variable, use this food or that food. Right. So make sure because I know for some people that part that's pretty straightforward, but some people still get that kind of spun around or confused. Make sure that that's clear, because that's at the core of everything else. So I would say, make sure you know that. All right, as we continue on. Whoops. Correlation, you should remember what a positive correlation or a negative correlation is. We've been talking a bunch about that. What kind of correlation are we seeing here with the cups of coffee versus time napping during lecture?
Negative, inverse.
Correlation.
That's a negative or inverse. As my independent variable goes up, my dependent variable is going down. Positive, if I was going to be like, the number of, the urge to poop versus the cups of coffee consumed, it would probably be a positive correlation. The more coffee you've drank, the more you feel you got to poop in the morning or something, right? Like, so you can have positive correlations. What if, what's another way? What's the other relationship we often find in science?
No correlation.
No correlation at all, right? So a lot of times you really hope there's some correlation, you're looking for it, but it's not. And I should also just make it, a lot of times in science, because linear relationships are so much easier to work with than more complex relationships, we always try to approximate things as a linear relationship. But most things in life are not linear relationships, right? Most things in life are, or they're linear, but only in a narrow range of where they're working. So don't just assume that the relationship is always going to be this pretty, but it can be a good first approximation, right? Like, if I, more and more coffee times, like I've mentioned, like if you're, I could do it on here. Cups of coffee. and exam scores. And it's like people who drink more coffee end up doing better, but then at some point they just get so jittery they can't even sit still. And it might be something like that. A lot of times relationships are like that. They're not just some beautiful, but if you say within this relevant range here, we have, it seems to be, it's kind of what we saw in our relationships with the enzyme lab, right? In the enzyme lab, if you do substrate concentration versus reaction rate, you get a nice linear relationship, but then at some point it becomes nonlinear as you saturate the enzyme and it starts flattening out. So just kind of putting that out there, real world relationships between independent and dependent variables are often complicated, but in general, we try our best to confine things to linear ranges because the math is so much easier.
So oh, sorry, just one quick question. So are you expecting like an answer to be like there's a positive linear correlation or just a positive correlation?
I mean, if I drew a line in there, you could say a positive linear correlation.
Mm-hmm.
Yeah.
Okay.
And if you've been spending time messing around in Excel, you've probably seen you can use many different orders of curve fitting under when it says add trend line.
Right.
Second order, third order. So, but anything we're doing in our class, for the most part, we are just messing with linear fits.
Okay.
All right, metric system. You should know the basic metric system. I would feel okay making sure how to convert between meters and centimeters or something. Because that is kind of a core thing in this class, and it's something that as I've been grading them, some people are good about units, some people are not so good about units, and also some people are not so good about kind of just that reality checking of units. So you should know the basics of the metric system and how that works. I would say that is important, not just for this class, but for science in general, or being out in a clinical setting where things are measured in mils. You should know what a mil is and how mils relate to liters and things like that. Or how grams, or you dose out drugs in milligrams and micrograms, and what is all the relationship between all of that. So here I said, is a liter bigger or smaller than a bread box? Can anybody answer that?
It's about the same.
Maybe people didn't, maybe you guys didn't. When I grew up, we always had a bread box in the house. Do people not have bread boxes anymore?
I have no idea what that is.
No.
Me neither.
You guys?
Same here.
Smaller.
But it can hold like
Yeah.
a couple loaves of bread, so it's like a liter smaller.
Yeah.
Right?
Oh my God. So you guys have never heard of bread boxes? Okay. Yeah, these things. Like, let me go to images of bread box. Like this, something like this. Yeah. So
It's about 2 liters, right?
it's way bigger. A liter is just like a water bottle. A liter is about a quart. So these are, so remember, so you guys didn't used to play 20 questions? That was always like the first question in 20 questions when you're playing 20 questions. Is it bigger than a bread box? Maybe that's like something that doesn't exist anymore. That was always, all right, let's go back. Oh, I need to get. We are not gonna worry about that now. Chemistry review. So what I'm gonna do now is kind of go through the different labs. And for each of them, I'll just, I'll try to like bring up the main points and maybe just quiz you a little bit and make sure that you're getting the main points. Like for the buffer system, what was the take-home message we saw in doing part 1 of the chemistry review?
Buffers don't really change the pH or don't
Or they
allow
keep it
for a
from
big.
changing too much.
Yeah.
Yeah, so they stabilize the pH. Right? We saw that pretty dramatically. And what was the evidence that how did we demonstrate how well they worked?
Did one experiment without a buffer and saw how adding acids or bases would change the pH, and then after, when you added in the buffer, how it mitigated any changes in pH.
Yeah, so exactly. So like, when you're thinking about these labs, make sure you remember what you did and what it showed you and how you knew it, right? We saw adding and also I would say right now, like, I don't know, I can't see exactly where people are writing, but I know like if we were in lab, a lot of times people are like scribbling notes in their lab notebooks. And it's kind of harder to see that right now, but I'm assuming that's happening to some degree here. So it's fine to take notes, but you should not be scribbling these notes right in your lab notebook right now. And you can use, if you come up with stuff as we're going through this and you realize you need to adjust or augment your discussions or something, that's okay. But you should not just, the lab notebook is not a place just to be cribbing notes. So just putting that out there. All right, so buffer systems were an important thing that we saw. Next, electrolytes. And actually the electrolytes and the carbohydrates, the one with glucose, those were basically two sides of the same experiment. What did we do? What did we find? What were some of the major take-home messages from all of the stuff we saw with electrolytes and organic the carbohydrates?
That
Carbohydrates
salts
don't dissociate.
dissolve and dissociate and form electrolytes, and some carbohydrates dissolve but not dissociate in water.
And what was the evidence? How did you know that?
Conduction of electricity.
Yeah, so remember we saw, and how did we know that it was conducting electricity?
Sound, but
Oh, yeah, the buzzer went off.
Yeah, the buzzer went off. So like all of that should be something that you're reviewing. Like, what did we find? How did we figure it out? What are the deeper implications? Like what's different about glucose and salt that made them act so differently when you put them in water?
Glucose doesn't dissociate, it just dissolves, so it's not a conductor of electricity in the same way as a salt.
And why does salt dissociate? What is,
Because of its bonds,
and what kind of bonds does it have?
ionic.
The ionic. So remembering the difference between ionic bonding versus covalent bonding, and all that. So you make sure that you are looking back. And I know, like, grading this one, some people were really good at connecting all the dots and seeing all the bigger pictures, and some people seem pretty like there's this thing happened and this happened and didn't really connect it as much about, well, what does it all mean though? And why did it happen? And how do they relate to each other? So make sure you really understand, what this showed us about, covalently versus ionically bonded things and how they are similar and different. What happens when you use water instead, oil, when you try to dissolve them in oil?
no
They
sound.
don't dissolve because it's not polar.
Right, so we had the whole, so did it conduct electricity?
Nope.
No. What if, so we never did it. What would happen if you tried to dissolve the sugar in oil? Would that have worked?
No.
Well,
No, why not?
because sugar does not dissociate, and that means the on- yeah, it does not dissociate into like individual ions. So ion has the ability to carry electrons
Right, so,
uh, discharged by the probe.
right,
Yeah,
It's not.
so let's go. I think, I'm not sure. So my question was, why does sugar not dissolve in oil?
it's
Because
solid.
oil is not a solvent.
I mean,
Because
Because
it's
but
like doesn't, they're not of the same thing. So like dissolves like, and they're different
polar, so non-polar solvent,
polar and non-polar.
right? So the sugar molecules, which are not going to become ions, but they are polar. They have charge imbalances that if you put them in water, the individual glucose molecules are going to want to make hydrogen bonds and go into solution. But in oil, there's nothing interesting. It's all non-polar oil molecules. So a polar sugar molecule is just going to stay stuck on another polar sugar molecule and stay in its little sugar crystal, right? So, so that idea of like dissolves like, polar and non-polar, I mean, we'll be seeing that till the very end. The last week of class when we're getting into the endocrine system and talking about steroid hormones and transporting them through the bloodstream, it's all going to come down to the same idea. Looking at how, why we have specialized ways to carry fats and cholesterols around in the bloodstream. So, a lot of this stuff is really important to understand kind of, hopefully kind of intuitively. So as we talk about, like, of course you need to have a carrier for this because the plasma is mainly water and the steroids based on cholesterol, which is a lipid, which is nonpolar. So la la la. Let's see. Where did oh the one looking at how come glucose looked different than starch when you looked at a jar of glucose solution and starch solution?
Glucose is a monosaccharide, starch is a polysaccharide, so it spreads the polysaccharide, disperses the light more, so it looks milky.
But there's something more that you'd want to mention.
It's a colloid.
Right. I mean, it's a really big polysaccharide, but it's a colloid. It's one of these things where you can force it into solution, but it scatters light. It ends up looking cloudy, but it never settles. All right, we did not do this. Well, I was very proud of coming up with this graphic. Yeah, that could be coronavirus. No. All right. Jumping lab. For the jumping lab, again, like all of them, you need to make sure you're clear about what the objective was, what were the independent, dependent variables. You should be able, you should know about, I could ask you about I could ask you, what was your hypothesis and your data supported or not? Any of these things, I could ask you more general things or generic things. I could ask you about somebody else's. I could say, here's some data from another class. And does this support your hypothesis? So you should, the test could bring in things that are more specific to your particular lab, and I have copies of all your labs, so I can see what your hypothesis was. You should know about be able to kind of intelligently discuss what were the things that we found. What did anything seem to be correlated? What was it? What were, blah, blah, blah. Osmometer. This one is crazy how this one kind of gets people confused. Um, the osmometer lab. There are still a few people who, despite all the back and forth and stuff, it's still not clear to me really understood what the objective was on this lab. What was the dependent variable? What was the independent variable? Does anybody want to say, so what was the independent variable? What was the thing that we were varying?
Concentration gradient, and then dependent variable would have been the rate of osmosis.
Exactly. This was the concentration gradient across the membrane. So again, we have this semi-permeable membrane here, the dialysis membrane. We have some gradient. There's some difference. There's some concentration inside. Outside was just, distilled water. And then there's some difference. What is the difference? Delta is just the change. What's the difference? What's the difference between the inside and the outside across the membrane? And then our dependent variable was osmotic rate. Like, how fast does water cross the membrane? How did we actually have a measure of the osmotic rate?
Measuring the amount that went up the tube.
Yeah. The more that water went up, it went up the tube. So we could look at the rate. How fast was it going up the tube? We'd measure the height at different time points. And how do we rate is another thing you should know about. How do I calculate rate?
Distance over time.
Distance over time.
It's distance over time. So if I know that it ended up going up 5 millimeters in a minute, it's going up 5 millimeters per minute. If it went up 10 millimeters in 2 minutes, how fast is it going?
5 meters a minute.
Say it again.
I think I got it wrong. 5 meters a minute.
So 10 millimeters.
Oh, 10 millimeters.
So it goes up 10 millimeters in 2 minutes.
So 5 millimeters a minute.
5 millimeters a minute. Right. Again. So all you do is what is the time that you're looking over? How far did it go in that time? And you calculate it and you got a rate. We're going to be looking at rates again later on in this class and some of the other labs. So you want to really make sure you understand what rate is and how to calculate rate.
So if it was 10 minutes 10 minutes every 4 minutes, you just divide 10 by 4 and that will give you the millimeters per minute.
Yeah.
Okay.
Right. And when we were doing this, we were kind of getting the average rate for 10-minute blocks, right? How far did it move in from 20 to 30? How far did it move from 30 to 40? And then we took that delta height and divided by 10 minutes to get the millimeters per minute for each of those, the average time for that 10-minute block. So we were kind of getting average rates for the different periods of time. Like how, right? Right, but it wasn't purely, a lot of people got confused. They thought it was about concentration being the independent variable, but it's not, right? It's the gradient, it's the difference across the membrane that is driving the osmosis. Like I could have 40% in the beaker and 40% in my little thistle tube. And what's the osmotic rate?
There isn't a change.
What's that?
Zero.
It'd be zero.
Zero.
No, if there's no concentration gradient, there's not going to be any osmotic net movement of water. So making sure you realize it's the difference on the two sides that's driving this. So what would happen? So I could say, so you know, we did put 40% in there and it started going up. And would it keep going on forever and ever?
No.
No,
Why is that?
because gravity pushes down on the water. There's a hydrostatic head.
Exactly. So as the system keeps going, gravity is just heavier as the column of water gets higher and higher. And so you've got a pressure pushing back on you. What if I had the tube sideways? Would it keep going forever?
Yes.
It ultimately would keep going forever, theoretically. But what would we notice? What would we notice about the rate as it keeps going?
The rate would slow because there's less of a concentration gradient over time.
Exactly. Right. The more water comes in, the less the concentration is inside, which means the gradient is going down. So from the moment the water starts crossing the membrane, we have less and less of a gradient, so less and less osmotic pressure. So if I looked at, let's say I let this go and it, at some point gravity is fighting and now it's hit some equilibrium. Whoops. Let's say it's hit some height where it's not going up or down anymore because gravity is just exactly the same, pushing back with the pressure that the osmotic pressure is pushing up. How do I know? If someone said, like, how do you even know that, you know, this permeable, semi-permeable membrane doesn't let sucrose through?
There wouldn't be a change. Well, there'd be no, which one am I saying? There would be no osmosis because the solute would diffuse out of the solution into the distilled water.
If the sucrose could move, it wants to. It's 40% in here. It's 0 out here. The sucrose would rather just diffuse out of the bag and just equilibrate. The fact that osmosis is happening and staying here is evidence that this sucrose can't cross the membrane, can't get out of the little bag here, right? What would happen if I put 20% sucrose in the outside here instead of just distilled water?
It would still increase in the tube, just not as much.
Yeah, exactly. It's gonna just have, it's gonna go up, but will it be faster, slower, or the same rate?
Slower.
It's gonna be slower because now we have, you know, half as much of a gradient. So it means our rate's probably gonna be half of what it was. And probably the height that it equilibrates is not gonna be as high. You don't have as much osmotic pressure, so gravity is gonna win out, you know, at a lower distance. So just kind of putting all this out there. Are there any questions about this? I know that osmometer and osmosis often kind of trip people up. And like I've mentioned, osmosis is going to be kind of upfront and pretty much to the end of the semester. The last couple of weeks of class are just going to be all about osmosis still as we get into functioning of the kidneys and things like that. So osmosis, you really want to just start feeling intuitive about osmosis and how it works and everything. Whoa, Oh, Marissa, watch out. The cow's about to like impale you on the horn there, I think. Let's see. Any questions about this one? Okay. The demonstrations of diffusion and osmosis, you know, a number of people got it and a lot of people didn't. What are you seeing when you see Brownian motion? We saw the little pollen grains dancing around in that little video on YouTube. Why are they dancing?
Molecules crashing between each other, like colliding. That's
Or I shouldn't, it's not so much into each other, but into the pollen, right? The pollen is floating on the water, and the water molecules, which are keep whacking it, and so the pollen gets all jiggled. Are you actually seeing molecules moving?
Nope.
No, you're getting kind of the secondhand evidence that they're moving. Diffusion. Yeah, what kind of things affect the rate of diffusion?
Temperature, surface area.
And so in which way? Tell me if it goes up or down. So tell me the thing and whether it increases or decreases the rate of diffusion.
Temperature would increase the rate of diffusion.
Yeah, temperature is going to make it go up.
Surface area would go up.
And more surface area to cross is going to increase.
Permeability would go up.
Permeability, the easier it is to go across.
Molecular weight would go down.
All right, that's what we saw on this one, right? The larger molecular weight. And some people got this wrong. They were just talking about, oh, it's like bigger or this or that at first, about distance. It's if you have a certain amount of energy and you are more massive, you just move slower. You know, you have the same momentum. But if you it's kind of when I was using a finger flick of energy on a paper clip, it flies across the room, and a finger flick of energy on a stapler, it barely moves, right? So the same amount of energy in something, if it has very low mass, it's just going to be zippier. Oh, this one. Again, people were kind of all over the map on this one. Definitely you should understand like how to interpret the test tube. You know, and some people did beautiful jobs of describing what they saw and drawing pictures and describing the supernatant. Was the supernatant red or clear? Was the pellet present or not? Were the pellets a little more compressed or not? Other people were really not that good at that, which is going to make it harder when you like kind of go back and like, how do I know hemolysis occurred or this or that? So make sure you kind of understand, like, what would it mean if the supernatant is red?
Hemolysis occurred.
At least at some level, some blood cells exploded because there is hemoglobin in solution. That only happens if there's hemolysis. If you can see a visible pellet, though, what does that mean?
That some cells didn't explode.
Combination.
Probably some of them didn't explode and got pulled to the bottom. If every single one exploded, you wouldn't see much at all on the bottom. Did we see any cases where you actually had a red supernatant but still had a pellet?
Yes,
Mhm.
in 0.10 molarity NaCl.
Yeah, and remember that with test tube 3, that 0.2 osmolar is hypotonic, but kind of weakly hypotonic. So some of them exploded, not all of them exploded. which is evidenced by the fact that there was a pellet, means there's still some cells, but it's red, meaning some of them died. So, so you can have incomplete hemolysis, right? And make sure there's still, again, from grading these things, some people still are not clear about crenation versus hemolysis. What does it mean to crenate?
Cells shrivel.
Shrivel.
The
Shrivel.
cells shrivel. What kind of a condition would result in that?
Hypertonic.
Hypertonic. So hypertonic, it's more osmotic outside. Water leaves the cell. Hemolysis means kind of the breaking apart of the blood cells. That's in a hypotonic solution. So the water comes in, and if enough comes in, the thing pops. So make sure you keep that all clear. I promise there'll be some questions around this kind of stuff. And it can get hyper, you start swirling around your head, and you're like on your brain's on some like merry-go-round, and it's like, wait, what is it? Up, down is up, what? So, spend some time making sure you're really solid about what is hypertonic versus hypotonic versus isotonic, what conditions lead to hemolysis versus crenation versus, happy cells, so, as you go through all the different tubes, make sure kind of what happened and why. And again, a lot of people were solid on this, so, but you should be able to know like what happened, why, how did you know, all that, why did it make sense? And again, we've had some weird stuff, like what was the osmolarity of the urea tube?
Be isoosmotic, but hypotonic.
So, and how does that work? How can you be isoosmotic but then hypotonic?
Because it penetrates the blood cell.
Right. So so make sure you understand that whole idea. We have a whole bunch of things. Basically, all of these are isoosmotic, and yet how they behave is very different from one to the next. So make sure you can understand what we saw and why it makes sense, and how the solutes are different from each other. And it's about both the concentration as well as the nature of the solute. This was pretty self-explanatory. What was the appearance of the model in the hypotonic solution when you looked at the little sausages?
Swelled up.
It was really swollen, right? Remember, it was one of those, like, in fact, when we do this in lab, we have to be careful. If we leave it in there too long, they actually pop on us. Where it says, explain an experiment that would regenerate the model after exposure to the 3 molar sucrose solution. What did it look like after it had been in the 3 molar sucrose solution?
Shriveled up or like flaccid.
It really kind of shriveled up, lost a bunch of water. How could you regenerate that little sausage, make it look back exactly like it started?
Put it back in a isotonic solution, so it's the 1.5 molar sucrose.
Exactly. If you put it back in a 1.5 molar sucrose solution, right, the water is gonna equilibrate until it's exactly 1.5 inside and outside again, and it's gonna look just like the control again. Right. So, hopefully that kind of makes sense. Enzyme lab. Again, this one, some people Some people really stepped up and got it, and some people seem pretty lost on this one still. Um, and at the beginning, we need to remember independent and dependent variable. What was the independent variable for this one?
Substrate concentration.
Yeah, it was just substrate concentration. You know, and this was something that we could easily manipulate. We either had, just, we had H2O2 and H2O, and we could just do these serial dilutions. We had 5, mils, 0 and just pure 1, 4, 2, 3, 3, 2, 4, 1, 5, 0. So basically, it goes from 0% to 100% concentration of hydrogen peroxide solution. So that's easy. That one was easy to manipulate. What was the dependent variable?
The rate of reaction.
The rate of reaction.
Yeah, so the reaction rate. This one is not so easy. How did we actually get measurements of reaction rates?
That we use the slope of the line.
Slope of what
For
line?
each concentration.
What was it? Milliliters over time, and then the slope of that graph was the reaction rate.
Yeah, so for each trial, we let time go. And then we captured the bubbles up in that burette, remember? You had to do 50 minus the reading because of it was all upside down, but let's just say mils of oxygen. And we saw something like that. As time went on, more and more oxygen's produced, which we can measure. And then I needed to get my best fit line. So you needed to take a ruler. Again, some people like freehanded. It's like, you can't do this freehand. So you put the ruler down. I'm actually putting it on my iPad here. And let me use a plastic. That's interesting. The metal ruler seems to be messing with my little Apple stylus. Totally does. That's wild. So I make my best fit line. I want to do it before the reaction is dying, when it's really rolling along. Why do I need to be careful about not dealing, being too close to the origin here? What did we find actually from looking at our control that's going on over here?
When you inject,
Because, oh,
oops. When you inject something into the flask, it pushes air into the burette, which screws up your results.
Right, because when we did the control, we just put pure water in, there shouldn't have been a reaction, and yet we saw 5 mils change, and we know that's not from the reaction. So we realized, oh crap, there is just a big artifact, this big bubble of displaced air that's going up here. So we need to make sure we don't figure this into our calculations, or it's going to totally mess things up. So starting at 5 seconds and on, and then before things actually go and start dying, draw a best fit line, and then figure out the slope, which is just rise over run, delta x over delta y. What? Good God. rise over run, delta y over delta x. And you calculate that out and you're going to get some mils of oxygen per second, and that's going to be your reaction rate. So a lot of people got this, some people didn't. You do this for each trial. What's the next thing you need to do though? Because this was just the way you go about getting these rates. What are we actually interested in? What was the actual objective for this thing?
What
To
happens?
What's that?
find a correlation between how substrate concentration and reaction rate changes. So then with the results, we made a graph.
So we want to see what is the relationship as substrate concentration changes, how does reaction rate change in relationship to it, which means all of these graphs, these don't tell you that directly. These are part of the process of getting to where you need to be. What we really want is a graph that has what for its axes?
Substrate concentration on milliliters of H2O2
Right?
on the X.
So we could say, let's say 100% and 0%, whatever. And then reaction rate, which is the thing we calculate for each of those different trials, which was mils of oxygen per second. You know, and then on this one, you just can start plotting it out. Like it was at 20%, at 40%, at 60%, at 80%, and see what happens. You know, and a lot of people who did go make it through this did find that as the concentration of hydrogen peroxide increased, the rate that the reaction went, how steep that slope was, how much oxygen made per second was actually going up. In this picture I drew here, I have it kind of flattening out here. What could this mean on this graph?
Saturation of the enzyme.
And explain a little more.
All of the enzyme binding sites are completely full of the substrate, and so the reaction can only go as fast as how the enzymes can turn over the.
Yeah, so we're adding more and more substrate, but they're going as fast as they can, so the reaction rate is still at some max. You know, where people get really confused is coming back here, where, you know, my time versus mils oxygen for a single trial, it also tends to go up in a linear fashion and then flatten out. But it's something really different happening. Why is this flattening out here?
The reaction's over.
Reaction's over. There's no reaction. The reaction, what is the rate at this point?
Be zero.
Zero. The best fit line is a flat line, slope zero. So make sure you keep that straight if that was something that was confusing. Like what is going on in these graphs of time versus oxygen that we use to calculate the rates versus the graph that we then make using that data that we can actually use to kind of look at our hypothesis and see if it's supported or not. Is there going to be any different if I have 60% and 80% and the rate is the same? Are they going to be exact? Is everything about those reactions going to be the same?
No, the one with the greater substrate concentration will produce more oxygen.
Yeah, this one will go on longer. Like if I was looking at it, time versus mils of oxygen, you know, if this was like, you know, 80%, it went like you know, and then I had like the 100%, you know, oh, it's got the same rate. But since there's more hydrogen peroxide, it's going to continue on for longer before it ends. I'll make more oxygen before I'm done. But in terms of the rate is identical, right? This would be with saturate. If I didn't hit saturation, what would it look like when I hit 100%? If let's say I was not at saturation yet, what would the next graph look like? Use words.
A positive, like straight line.
No.
Like the line would continue.
Say it again.
The line would continue.
No. So how are we assessing rate in all of this stuff? What is the thing that's related to rate?
The slope of the line.
Slope. So if I am not at
Oh, that's
saturation
right.
and I'm
The slope would
increasing,
be
The slope would be steeper.
slope would be steeper. I would have a steeper slope if I'm not at saturation, because that's what faster rate looks like, a steeper slope, right? That's the whole point here. If I hit saturation, the slope is the same because it's at the same rate. If I'm not at saturation, it's a steeper slope because the reaction continues to speed up with higher concentration of substrate. So is that clear? Is there any questions about that? Like if what I just said is making you like, why, what? Then please ask a question, because otherwise you're going to miss something on the, on the exam. This is kind of making sure you get this relationship between the two different graphs and between how we measure reaction rate and how reaction rate ends up manifesting graphically and all that.
I'm a little confused just at that end part in terms of you saying just what the graphs would look like of the difference. Like rates are equal, but O2 is being produced in the 100% one would be that it would just continue versus that the slopes would be different because the
Yes.
reaction rates are at the same rate.
So again, if this is second, this is time, this is O2. I know, let's say again, I was using different colors. Let's say 80%. Let's say just looks like this, right? It's going to go up at some certain rate, which is going to, you know, be some certain slope, how many mils of oxygen per second are getting made. And then if I add in the next one, it's again like 100%. There's a couple of things that can happen. If I'm at saturation, what is the slope going to be as this thing starts?
It would be the same.
It'd be the same. I'm already at my maximal rate, so I'm not going to go any faster. So the mils of oxygen per second being produced are going to be the same as in this previous case. But since there's more hydrogen peroxide in the mix, it's going to keep going a little longer until it's all broken down and it's finally done. I'll make more oxygen before it's done, but the rate at which it's happening is the same as I got at the 80%. You know, if I drew the graphs for these and made my best fit line and calculated the slope, they're the same number. You know, again, so this would be at saturation. You know, if I had 100% and there was no saturation, meaning I'm just still in this linear thing where you add more substrate or you increase substrate concentration, increase rate. Now what I'm going to get is it's going to go up faster. You know, it's, whoops. It's going to go up faster. It's ultimately going to make the same amount of oxygen. By the time it's done, you will have, the exact, however much oxygen you make when you break apart that much hydrogen peroxide. But the rate that it got there, it made that oxygen faster. So does that make it clearer?
Yeah, thank you.
Okay. 1134. So moving right along. So nerve. Well, we came back at 10, so we've been going for, let's take another little break, and then we'll continue with nerve stimulation. I was realizing it's been a while. So it's 11:35. Let's come back at, I don't want to run out of time. So let's come back at 10 to noon. So 11:50. 11:50, we will regroup here. So we are going to continue with our review of the lab material. When we last left our review, we were about to hit the nerve stimulation lab. So let's continue there. All right. So I think, as always, it's important that everybody gets what was the independent variable and what was the dependent variable in this one.
Location on the body and frequency of stimulation were the independent variables.
So this one we did. Right, we tried a couple of places. We looked at the finger, we tried at the calf, and then the frequency of the stimulation. We had single pulses, we had 1 pulse per second, we had 5 pulses per second, 2 pulses per second. So that was our, there were 2 independent variables. What about the dependent variable?
The perception threshold measured in milliamps.
It was like, what was the minimum amount of electrical current that you would put on their skin that they would just barely feel as that was something. I felt that.
0.1.
Right.
No.
So, and what were some of the hypotheses? And again, there's 2 relationships. One is location, and the other is frequency of stimulation versus perceptual threshold. What were hypotheses here?
Areas with greater sensation would have a lower perceptual threshold.
Why?
Because they have more sensory receptors, and spatial summation increases the graded potential to initiate an action potential.
Right. And so again, one of the things that we've been, and this actually tied in nicely with the general sensation lab, this idea that you have receptors under your skin that are the things that convert the actual stretch of your skin into an electrical signal. And some parts of your body, like your fingers, have a lot of these little receptors underneath. Other places like the back of your calf, oops, they are not nearly as densely. Again, remember we saw in the back of the calf, right, the later lab, you could actually have two pins that were up to like an inch apart. And you wouldn't even know that it's two versus one pin. Whereas on your finger, if they're an inch apart, you so know it's two pins and not one pin. And it's because of how kind of densely arranged the little receptors are. Have more of these things going across, there's more likely you're gonna, a lower voltage is gonna like send enough messages to the brain that you actually notice something. So what about the other independent variable?
And increasing the
greater the
frequency of stimulation will decrease the perceptual threshold
And again, we always need the why.
because of temporal summation. So as the frequency of stimulation adds up, then it will create temporal summation, which will decrease the perceptual threshold.
Right, so you don't need, if you can be completely just make up these axes. Like let's say the overall voltage has to get to here for you to feel it. And if you just do a single pulse, it doesn't get high enough. At this high, you have a single pulse, you have to make the pulse that big to get there. But if I use a little pulse, but then I do them in quick succession, they start adding together. So they kind of dogpile on each other and we actually do hit that threshold. So the individual pulses don't have to be as big if they're coming in quick succession. Because they start adding to each other. Right, so, and what did we find? Was that, did the data support our hypotheses?
Yes, the increase in frequency at a lower perceptual threshold.
Yeah, it was pretty clear. Yeah, we saw on both on the finger and on the calf. So this one, but again, this is another one of the ones where it's just really important to make sure you understand what we were looking at. Like the people that had trouble with this just never wrapped their head around what are we even looking at? What are we varying? People would like, how does the resistance and the voltage or this, it's like, no. It's like we're looking at different areas of the body. We're varying the frequency of the stimulus. And this is another one of the labs where measuring the dependent variable was kind of an ordeal. How did we actually measure this dependent variable? What was the process?
By measuring
Or
the current when they felt something.
the stimulating bar electrode, it is.
But kind of back up a bit. Where do you start?
Start with a very low current and then just keep increasing it by a certain amount until the subject reports they can feel the stimulus.
You have to start at a level that is below what they can feel. The only way you can actually figure out the threshold is to start below it. Right? And then you can kind of increase it. And when you can find, okay, that's the line. That's the line between can't feel it and can feel it. And that's the threshold there. We're going to be looking at different versions of thresholds as we go on and just noticeable differences. Weber's law is actually the same Weber who came up with the Weber test with the tuning fork. also did a lot of cool seminal work on just sensory perception and like how can you tell the just noticeable if you're holding two balls, and are they the same weight or is one heavier or lighter or which one is which? That's another threshold. What is the minimal difference in weight between two things where you can just tell that they're different versus I can't really tell for sure if they're the same or they're different. That's another threshold, the just noticeable difference, they call that. So there's a lot of these things in sensory neuroscience that are really kind of fun, kind of mapping out the limits of what we can detect and what we can discern. And again, we saw it again with the 2-point discrimination and things like that. And again, we saw a nice overlap between the 2-point discrimination and location of the body and nerve stimulation. Fortunately, this is what you like. You like in science when the results kind of reinforce each other, right? It would be really a drag if the fingers were most sensitive for the nerve stimulation lab, and yet they were horrible at 2-point discrimination compared to the calf. It's like, oh crap, like this is not making sense. I need to go back and revise my whole hypotheses and understanding of what's going on here. But luckily, it's all consistent. The fingers were indeed way more sensitive to touch discrimination as well as this nerve electrical stuff. So, nerve stimulation lab. Reflex action. What was the independent and dependent variable for the reflex action lab?
I think it was angle of knee extension was dependent variable, and then doing it with and without gendrastics was the independent variable.
Yeah, so independent was with or without gendrastics. And the dependent variable was maybe a little more general than that even, because we looked at it in a couple of situations.
Was it the reflex itself?
No, the reflex was what we were working with, but it was something related to the reflex.
The magnitude of the reflex?
Exactly, the magnitude of the reflex. So in the case of the patellar one, it was the angle. In the case of the Achilles one, it was just kind of a qualitative thing of like, did they kick out? Did it look harder than it was before? Right. What's the difference between qualitative and quantitative?
Quantitative has a numerical value.
Yeah.
Qualitative is quality based.
Yeah. So, when we measured the actual angles for the patellar, that's quantitative. But we could also, oh, hold on. Sorry. Oh, shoot. All right, so back to, sorry, I just got distracted. My reflex action, let's continue on. Actually, what did we find?
You were just saying how the patellar measuring angles is quantitative, I think.
Right, and then what did we find for the results?
With the gendrastics, it increased the magnitude of the reflex.
Yeah, which was consistent. And I don't know if you, I was happy to see most people kind of addressed the data. Like when you look at the data that we had across, our quote unquote class, I think it was an important thing. I made sure I left the subject in there that did not have that result. Because I really think it's important for you to realize that real data is messy and that not every subject is going to behave the way you expect or follow your hypothesis. And what you need to do is, over a large number of people, find out on average, does it seem to support your hypothesis? I mean, I think it's also useful just when you're in a clinical setting, realizing that every individual is an individual and might have an idiosyncratic reaction to something that's not what you expect. And that isn't typical. That doesn't mean you should throw out your expectations, because in general, this is, this or that is what you see. But this idea of this idiosyncratic reaction, it's important to keep in mind, realizing there might be somebody who hit their knee and there is no kick at all, or somebody, for who knows what reason, there actually is nothing wrong with them, but they're just acting, they're responding in a way that's not as typical. So just kind of remembering that, we're looking at, kind of general, we're looking at trends, measures of central tendency, not necessarily like we are defining what we're going to, how everybody's going to react. We're going to say, in general, this is what you tend to see. Boom, boom. These guys, yeah, you should know what, you should remember them. Plantar, corneal, pupil. I was kind of bummed. There was a few people that totally missed, like people got all confused. I would say you want to stay focused, focused on the eyes. If you shine a light in one eye, what happens in the eye that you're shining the light in?
It constricts.
The pupil constricts.
The pupil constricts. And so even the word, like, make sure that everybody, you know what constrict and dilate mean. Constrict gets smaller, dilate get wider. Sometimes I'm not sure if people just don't understand what the words are, or if they're just mixing up the concepts. But constrict is getting smaller, dilate's getting larger. And if I shine it in my right eye, what's going to happen to my pupil in my left eye?
It also constricts.
It also constricts. We have that consensual reflex. So that's important. That's important on this one, that it's, again, we talked some reflexes just stay on one side, like the, that patellar tendon, you whack the right knee and the right knee kicks. You whack the left knee and the left knee kicks. You shine a right in the right eye and both the right and the left eye constrict. So again, depending on how these reflexes are wired, you can get different kinds of responses. Why do we even do all of these reflexes in general? Why are these a thing?
To test signal transduction or synaptic pathways.
What's that?
To test CNS or peripheral nervous system.
So it's usually to test the nervous system. It's usually, and not just the CNS, it's also to test the PNS, making sure that the nerve conduction is right, that signals are getting where they need to get and coming back to where they need to go to. And so a lot of these tests are for looking to make sure the nerves are working properly, making sure the processing, the integration is working properly. So again, these are really important parts of neurological exams. La 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 la. Keep going. 2-point touch discrimination. So we've talked about this a bunch. I should just, because a couple of people wrote this, I'll just mention it. Some people, when we, like, why is it a much more, like why are the lips much better at 2-point discrimination than your calf?
Greater
Because
density
there
of
are
A lot
receptors.
more receptors in the lips.
Yeah, more dense arrangement of receptors there. Some people were like talking about they're closer to the brain and spinal cord, so the messages go faster or something, which has nothing to do with that. You know, your messages are all, you just kind of assume they're getting where they need to get. There is an interesting thing where your brain actually does make corrections for the timing differences. So the people who were worrying about the distance from the brain, you're not like totally like off. There is this idea. It's kind of, well, let's, oh my God. All right. So this idea, here's your brain. Here's a receptor here on your lip. And here's a receptor on your toe. The fact is, it's true that the signal from your lip is going to get to your brain quicker than the signal from your toe, even though the fat myelinated fibers are really fast. We said over 100 meters per second conduction velocity. It actually is a real amount of time that it's going to take it to get up here. So what that means, if you touch somebody's toe and you touch their nose at the same time, the signal from touching their nose will reach the brain before the signal that came from the receptor transducing the touch at the toe. But you don't feel that. Your brain makes the calculation of when must it have started. This is where I'm not going to get into it that much in this class, but this idea of time, this flow of time, just like with all these other things, visual illusions, auditory illusions, you can totally mess with your experience of the flow of time as well. What happened first, what happened second. You can do these experiments where you stimulate, like if you stimulate this nerve at the same time that they see like a flashing light, like here's some flashing light, which is going at the same time that this nerve goes off here, they will feel someone touching their foot before the light actually flashes, even though the nerve is getting stimulated at the same time as the light is flashing, because their brain has to backdate the signal. Well, it's like if the signal is here now, but it must have taken it a while to get here, I must have gotten touched like a fraction of a second ago. So you actually, your conscious experience is like felt a touch on my toe and then I saw a flash. Because your brain actually backdates the information coming in to make, so your experience of the flow of time makes sense for what it's imagining it must have happened given the mechanics of your nervous system. So it's kind of trippy. There's a bunch of other stuff. There's really, again, if we were in a classroom, we could even do things with students where you can totally show how your experience of the flow of time, what happened first and next, can totally be manipulated, like pretty simply. So even things like time is, I mean, some, whether how long things seem to take or whether they seem to, it's like you set a timer for 25 minutes and damn it, it's gone already. Other times it's like, oh my God, it's like the clock isn't moving, right? And so there's that aspect of time, but then there's even like what happened first and second you can mess with. So it's kind of wild. So let's leave this back. So just talking about density of receptors, referred pain. Oh, wait, did I? Oh, adaptation. So what is adaptation first?
It's the ability of our body basically getting used to the stimuli, so we don't no longer feel it.
Exactly. So you have a constant stimulus and you kind of stop responding to it. And then if something changes, usually the receptor wakes up. What were the two examples of this that we saw?
The coin on the forearm.
Yeah, the coin, touch receptors. If you have a coin on your arm, eventually you don't feel the coins anymore. What was the other one?
The temperature adaptation.
So the temperature, and this is some people were great at their descriptions of what they did, and other people, like, definitely as we go on, I'll make another little like talk about kind of what works well. But if you put your hand in, some people were very good about what did it feel like. Um, when you put your hand in warm water, it feels warm. What happens after a couple of minutes?
It gets used to the temperature, and it doesn't feel as warm as it did initially.
Exactly. And then if you put your other hand in, what does this hand feel like?
It feels warmer.
Yeah, it'll feel warmer. So this one adapted, it's been in that temperature, but now it's just kind of like, kind of flat. But this one, which has gone from the air into the warm water, it's like, oh no, it is warmer than it was. And we saw the negative after images too, where we did the one where we had the three, you had the hand in the cold, the hand in the hot, and then you put them both in the lukewarm. And I know at least a lot of people had that experience of the negative after image of the one hand feeling hot, one hand feeling cold. And it's, again, I love doing that just because it makes you realize how your experience of the world is not quite as solid as you might think. Yeah, there's one, I went to one talk, it was great. It was about this, he was saying, there's a kind of a classic, there's a classic illusion where you have like two arrows or two lines. In one line, it's like that, and the other line's like that. And I didn't draw it that well, but in general, most people tend to see this one as longer and this one is shorter. And then you like, put these on, you realize, oh my God, my vision, I was getting fooled and this and that. He was like, your sensory system is constantly making mistakes and distorting things or misjudging things. It's just most of the time you don't have the little lines and arrows to let you know what the actual ground truth is on these things. I well, let me make sure I finish. Maybe next time I'll show you something I showed Stephen last time, like around the idea of just extracting pigment. What is the actual color of things, even though actually in reality, because of light and shadow, things look really different. There's all sorts of things that are really tricky to actually, even if you know what the real truth is, it's really hard to see what's really there. Let us continue. Referred pain. What is referred pain?
When you perceive pain in an area that's not receiving the stimulus.
And Why do we care about it? Why is it useful to think about?
Clinically relevant, so that if people are presenting with, there's certain pathways where it shows up in different locations in the body if internal organs are experiencing pain or damage, and so it'd be helpful in a medical setting.
Yeah, so it's very helpful because again, you can localize like pain on your body really well. It's like, ooh, I got a dart in the neck. But if there's pain in an internal organ, it's just, ooh, something hurts. But it's really hard sometimes to know exactly what's up. And the associated referred pain can help kind of diagnose what's going on there. So I mean to be, I don't mean to be, let me share something more interesting with you all. Well, now we're almost at the end. You know, the hearing. So most people got, what I expected. Some people didn't, and it could just be the subject was really good, or I think more likely maybe just they use other clues, people moving around them. In general, if you are snapping around, where are people the worst at localizing? Where are they more likely to not point exactly at the snap?
Along the midline.
On the midline. You snap in the front and they point up, or you snap over their head and they point forward or something. Why does that make sense that it's harder to locate a snap on the midline of your body than on the left or the right?
Because the sound is going into both ears at the same time and same magnitude.
And what are the implications of that? Why would that make a difference?
If it was unequal, so if we heard it more on one side, then we would be able to determine that it's in a certain direction. But if it's equal, then we can't determine what direction the sound is coming from.
Yeah, so one thing equal in, so one thing is that intensity difference. If it's louder on one side versus the other side, it's probably on that side that it's louder on. So what we call that intraoral intensity difference, the volume difference from one ear to the other. What other clue can you use?
The intraoral time difference?
Yeah, the intraoral time difference. Time between, your ears are actually, or your nervous system is actually able to tell the slight difference in time because sound is pretty slow. You can actually tell like, oh, it hit this ear before this ear, so it must have been over there. It was closer to this ear because of just the time it arrived. Whereas if it's something right on the midline, it's going to hit both ears exactly at the same time, whether or not it's in front of you or on top of your head or behind your head. So you don't have such slam dunk information. It's like if it's on the side, it's louder here, it's hit this ear sooner. So it's got to be on that side, right? So you don't have as much information when things are on the midline. Frequency range of hearing. Well, I can't show here. Let me. So we did not get everybody, but I got at least some people. Because not enough people actually put this in, I did not post it and I just let people use the data from the last class. But this is our class. This is the data people were emailing me. So you can see this real clear correlation between age and frequency. It's just unfortunately the way it is. Yeah, I mean, and don't take too, some people, I mean, some people wrote, oh, my hearing is 20 years old. It's like, no, it's a frequency, and in general, according to that guy who made the video, someone this age is usually around that frequency, but you should just think about the frequency. It depends a lot on, some of it's genetics, a lot of it is your, what you've been listening to and how loud, and if you've been, yeah, I mean, in general, the older you get, the more you like start wearing earplugs to loud concerts, because it starts getting scary to think about losing your hearing more and more. And there are a lot of really good earplugs now that don't change the quality of the music that much. They change more the volume. Like the old earplugs, those little foam things, make everything feel like you're listening to music through like a bunch of sponges. It's just boom, boom. But some of these, they're not that expensive. For like $20, you can get these really nice. There's Etymotic, there's different brands that make really good earplugs that cut across all the frequency. So the music actually still sounds pretty darn good, but you don't feel like this in the back of your mind, I'm trading off this moment of music for damaging my hearing. So public service announcements for somebody who used to kind of be an idiot about earplugs. I think they're good, especially if it's really loud. If it's not that loud, it doesn't matter. But there are places that play music really, 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 really loud, and that is going to accelerate this hearing loss. So
it happened to me, Professor. I did terrible in that test. Actually, I knew that I had hearing problems, but I didn't know that they were that bad. And it happened after a concert. It was super loud, and that was in February. And after that concert, I had tinnitus in both ears, but louder in the one that had the less result in frequency in the test.
yes, I mean, at this point, if there's quiet music, it's the same volume as the ringing stuff. It's just never, it doesn't ever go away anymore.
Never, but well.
So public service announcement, protect your ears because you only get one pair. And the hearing loss, again, this is another thing where some people did awesome. I loved how people were able to, like, you had 3 patients and talk about what the different tests, what the tests looked like and then what it means. Other people got kind of all confused about it, or some people didn't even they did one patient but not the others or something. In all of these, I promise you, there'll be something about the Rene and the Weber test on your exam. If somebody puts this little, becomes like the little unicorn here with what would it mean if he hears the tuning fork equally in both ears?
That he either has normal hearing or he has sensory neural or conductive hearing in both ears, or sorry, hearing loss in both ears.
So either everything's fine, or it could be possible he has hearing loss, but it's exactly matched. So it gives you some information. And then the Rene test, what would it mean if you put it on the mastoid, and then when he couldn't hear it there, you then put it next to the ear and he heard it again for just as long?
Normal hearing.
Yeah, that would be normal, because normally the air conduction should continue on past when the bone conduction stops. What if he didn't hear it anymore on the bone, and then you moved it here and he didn't hear it at all as well?
They would have conductive hearing loss in that ear.
Exactly, conductive hearing loss in that ear. So that's you should understand that. If he had conductive hearing loss, in his right ear, what would the Weber test look like?
It would sound louder on his right ear.
It sounds louder on the ipsilateral ear. If he had sensory neural deafness, it would be the opposite. Yeah, so make sure you understand these tests. Okay, I need to go much faster because I have 0 minutes left to get through the rest. Vision, blind spot, you should know what the blind spot is. You should know what color blindness is. Visual, I will spend just a few moments. Some people did not answer this the way I wanted. If you are 20/30 vision, what does that mean? Not just like, oh, you're worse than perfect. What does it really mean down to the numbers? What do those numbers actually mean?
And
That go, Lauren.
oh, I was going to say someone with normal vision can see something from 30 feet away, and someone with 20, well, then the other person can see it. Or, never mind. I can't phrase this right.
Lauren,
Well, if someone sees at 30 feet
tell
with
me what
normal
that says
vision,
about Lauren's vision.
we see at 20.
I can see something at 20 feet what someone else can see at 30 feet clearly.
Yeah, so you're standing 20 feet away and you see with the same acuity as somebody 30 feet away. So that means normal vision person can stand quite a bit further back and still see just as good as you. So your eyes aren't as sharp. And some people, and I don't understand how, that printout I think said 6 feet away, but a lot of people were like, I standing 10 feet away from it and my vision was stinky. And I'm like, it's because you're supposed to be standing like half as close. Anyway, so you should know that astigmatism, near point. Oh, and then finally, let's talk about this. It was kind of fun to see what kind of foods people's tasted and stuff. It's kind of making me think in the future to kind of mix things up in the in-person lab even. I do want to talk about this as a class before we cut it off because there were very different experiences across the different write-ups. Some people wrote things up that were very similar to what I normally see, but a few people had pretty atypical results. So I want to make sure that you hear the bigger picture. Does anybody want to share their experience of doing this?
So for me, the majority of the items that were tasted weren't really able to be identified until the person was able to smell again.
Yeah, so that's very typical. You put it in your mouth and it's like, I don't know what this is. This a carrot, a jicama, who knows? And then it's like, you start chewing it and then you've got like this, it's sweet, it's a fruit, but is it a plum? Is it a prune? Is it a, then finally they open your nose and it's like, oh, I know what this is. So that's very typical. Sometimes with just the taste and the texture. I saw some people had these kind of super subjects who you just put it on their tongue and they knew what it was without any taste or smell or anything, just pure feel. I mean, it might be they just are so used to the texture and the feel of it that it doesn't even matter what it tastes or smells like. It just has to, it's like, oh, that exact amount of squishiness or that exact amount of roughness or smoothness, I know that is an apple versus a pear, or that's a cheese versus a this or that, or that's cheddar cheese versus Swiss cheese.
Yeah,
but
that actually happened to my subject with apple.
So, I mean, so that's possible, but it's not as typical. But so when you think about what the experience of food is, what kind of things are actually, when we say, oh, how's food taste? What are we really talking about?
Smell.
So a big part is smell. Definitely. What else?
Texture.
Yeah, that texture, that mouth feel. The feel is also, for a lot of people, the feel was like one of the giveaways. So the feel is important of it. When you think about taste, you don't usually think about feel, but then, some people don't like, I have a friend who won't eat oatmeal because it feels like he's eating snot. Sometimes just the texture can be so intense that it trumps everything. And then taste is important, the sweet salt, but again, that's just one piece of all these other things. So that's one of the take homes. I like part of the reason I wanted you to do this at home. Partly it's fun, but partly I think if you haven't really thought about it deeply, realizing that what the food tastes like is something way deeper than just taste. And there were other things, we didn't get into like picante, like salsas, like Chilean things like that. That's a whole other thing. That's capsaicin, these other receptors that are binding and are creating the sensation of heat and stuff. So there's more than these are, this is a lot of it, but it's not all of it by any means. The of peppers and stuff is a whole other thing with its receptor opening up channels on cells. So, are there
Professor, one question. Sorry, this is just curiosity. Spicy, it's related to heat. I heard that it was pain receptors, or that was,
there's capsaicin? So we call it heat.
okay, capsaicin. Okay, thank you. Just curiosity.
I'm thinking if I'm adding some heat to this dish, I'm going to add in a little bit of chili powder or something. Yeah, no, I was, yeah, and I don't think, I don't know. There might be some cross-modal stuff there, but I don't know for sure, actually. So we've kind of gone through all of this. Are there questions about any of the labs or expectations about the lab exam?
I was gonna use marshmallow because I saw
Well, not yet.
unit next to
Um, so then I guess, I'll be here for the next hour if you have questions about anything. But otherwise, it's time to go off and do whatever you do on your Tuesday afternoons. I'll see you all on Thursday morning.
Thank you, Professor. See you Thursday.
Thanks, Professor.
Thank you.
Thank you.